Hello and welcome to Multimodal Techniques for High Content and High Throughput Cellular Phenotyping, presented by BioCompare and sponsored by BioLegend. I'm Tamlin Oliver, Managing Editor of BioCompare, and I'll be the moderator for today's event. Our speaker today is Peter Smibert, Senior Manager at the New York Genome Center. Peter will provide an introduction to a suite of technologies that uses DNA-tagged antibodies to couple protein detection with single-cell RNA sequencing. He'll review CITE-seq, cell hashing, and related technologies, and then explain how CITE-seq compares to other single-cell protein detection methods. Before we begin, though, I want to go over a few webinar logistics. We'll hold a live Q&A session at the end of the presentation. To ask a question, click on the Ask a Question tab on the right side of your screen. The right side also features an overview of this webinar and more information regarding our speaker. If you have a technical question or problem with the webinar anytime, click the Test Your Connection button under the presentation window, and we'll do our best to assist you. Finally, we encourage you to use the social media widgets at the bottom of the screen to share this event with your colleagues. I think we're ready to get started. Peter, take it away.
I'd like to begin by thanking BioLegend for the chance to present today. I'm going to talk to you today about some of the multimodal techniques for high content and high throughput cellular phenotyping that my lab has been working on for the last couple of years. I just want to begin with a disclosure related to IP surrounding this work. And so where the field is currently at the moment in terms of single-cell RNA sequencing, it's been an incredible decade of growth in this space, where we've gone from almost the first single cells being sequenced about 10 years ago to the point now where many hundreds of thousands of cells can be assayed in a single experiment. And you can see from this plot from Valentin Svensson that the growth in terms of the numbers of cells that are assayed in an individual paper, it seems to be increasing almost exponentially. A large proportion of this growth has been driven by technology. So showing in the top section of this slide, you can see the different methods that have been used to isolate single cells for capturing their RNA contents. And really, the growth that's occurred in the last 4 or 5 years has largely come about because of these methods on the right-hand side, these nanodroplet-based or pico-well or in situ barcoding-based methods. What these have in common and what distinguishes them from the methods to the left of them is that you lose addressability. So once you move into these types of methods, you no longer can retain information on phenotypic markers of the cells before they got partitioned into their droplet or their well or started accumulating in situ barcodes. So you lose information on cell shape or probably what is the most commonly used way of phenotyping cells, which is to look at cell surface proteins. So this gets into a comparison between single-cell RNA sequencing and I guess the current state of the art in terms of really high throughput single-cell phenotyping, which is using well described protein markers on cell surface to categorize cells. And this is done by flow or mass cytometry. And these methods are very similar to each other, but the basic idea is that you can detect proteins on the surface of a cell, and you get extremely robust detection of a few markers. However, by definition, it's biased. You only detect markers for which the reagents you put into your assay are for. And multiplexing is limited by the available number of fluorophores or metal isotopes, or just the ability to discriminate different fluorophores and metal isotopes from your instrument. In contrast, single-cell RNA sequencing is used to detect transcripts, and you get a sparse detection of many markers, which in theory is unbiased. So any poly-A transcript can theoretically be captured by these approaches, especially those that use the poly-T reverse transcription primers. But of course there's trade-offs. There's also very high rates of stochastic dropout, and you just don't capture every molecule. And also RNA can be an imperfect marker. It's the proteins are often a more terminal marker of gene expression that are more reflective of the particular cell state. So our goal was to see if we could figure out a way to couple protein readout with high throughput single-cell RNA sequencing and essentially generate something that was the best of both of these worlds. So this was how CITE-seq was born. So this is the brainchild and the work of Marlon Stokius, who is formerly of my group. He's now working for 10x Genomics in Stockholm. And he had the idea that you could couple protein detection to single-cell RNA sequencing by putting a DNA oligo onto antibodies and using that, allowing that oligo to be captured by the RNA sequencing method. So the oligos that he attached to antibodies look something like this. There's a poly-A sequence at the 3' end that enables these oligos to be captured by poly-T barcoded primers in the single-cell methods. There's an antibody-specific identifier, so a barcode sequence that tells you which antibody this oligo has been attached to, and then an amplification handle that you can use for subsequent library production and sequencing. Two really key points that need to come across here is that the barcode space is essentially infinite. So in a very short stretch of nucleotides, you can make more barcodes than you're ever going to have antibody reagents. And also, you theoretically have unlimited multiplexing, because you can design these barcodes with sufficient edit distance between them, and everything is being sequenced. So it's a digital readout. There's no danger of fluorophore overlap or anything like that. You detect this sequence in sequence space, and you should be able to put as many into one reaction as you like. So you're not limited in the same way that flow cytometry and mass cytometry are. So this is how a CITE-seq workflow looks. So again, your key reagent here is this antibody with the oligo. You stain cells with these oligo-labeled antibodies, and you wash to remove unbound antibodies, and then you encapsulate into a droplet. In this case, shown something like a DropSeq or a 10x genomics method. A proportion of the time, a single cell labeled with decorated with oligo-labeled antibodies will become co-partitioned with one of these microparticles that bears cell barcodes. When this happens, the cell will lyse, and the cell's mRNA content and the oligos that were associated with the antibodies bound to the cell surface have a chance to associate with the poly-T primers on the surface of the microparticle, or released from the microparticle, as the case may be. You can then follow standard library prep procedures. Ordinarily, when you do these types of methods, you size select your resulting cDNA. You keep the large fraction, which contains your mRNA-derived cDNA, and you discard the smaller fraction, which contains primer dimers and a bunch of mess that you don't normally need. However, in our method, we actually recover all of the useful protein information from this fraction that you would otherwise throw away. And this is important because it means that you're not sacrificing any of your complexity of your mRNA-seq component of the library in order to get the protein information. And this is just an illustration of what the two different libraries look like. They're very readily separable by size. This is the cDNA part of the library, and this is the protein or the antibody-derived tag or protein tag part of the library. So this is an example of CITE-seq on human blood. So this is just a t-SNE representation of 8,000 cord blood mononuclear cells that have been clustered based on their RNA gene expression. And you can see all the regular clusters you'd expect to see: T cells, monocytes, NK cells, B cells, etc. But we actually have a protein panel in this experiment as well, and some of the markers that were in this panel are shown here and sort of superimposed upon the clusters in which you'd expect to see their expression. And indeed, this is what you see. So if we look on the right-hand side here, the blue representation is the mRNA for a particular gene or protein, and then the green is the protein tag levels. And you can see a few things. So let's say for T-cell markers here, so for CD3, CD4, and CD8, you see very robust detection of the protein in the T-cell clusters. And fortunately, you see the anti-correlation of detection of CD4 and CD8. They're mutually exclusive cells. CD8 is also expressed in NK cells, as is an NK cell marker CD56. We see CD19 in B cells, a couple of different monocyte markers here, CD11C and CD14, and we see CD34 in the precursor cells. You can also see that mRNA and protein do correlate really well at the cluster level, as you'd expect. Of course, at the individual cell level, this correlation depends on the marker, but is generally relatively poor, largely because of dropout. You don't detect every mRNA molecule in every cell. Also, proteins are expressed at much higher rates per per unit than their cognate mRNAs. It's estimated between 100 to 1,000 fold more protein molecules than cognate mRNA molecules. So the other thing is that you can see signatures of potentially post-transcriptional regulation here. So if you look at CD4 example, in both monocytes and T cells, it seems to be detected at relatively similar levels in terms of the number of cells that the transcript is detected in. But the protein levels seem to differ quite markedly between these two cell types. So now I just want to show how CITE-seq can resolve some subtle transcriptomic phenotypes. So some of the more eagle-eyed people might have noticed on the previous slide in the CD56 panel, we have nice protein expression in NK cells, but there's a sort of lobe off to the bottom right, which I can highlight here, where the protein expression is higher. And this is something that's been known for a while, that there's populations of NK cells that are CD56 bright and dim, harkening back to the flow cytometry days. And this is kind of what it looks like. If you look at CD56 levels over these cells, you can see a clear peak of the dim cells here and a clear peak of the bright cells here, and cells where it's off. And what we see is that there's this inverse relationship between cells that are high for CD56, shown here, and CD16. And this is a known relationship in these cell types, and it's really pleasing to see that we can recapitulate that. And if we take these cells and take partition the cells into CD56 bright or dim, you can see that the dim cells have much higher CD16 protein levels than the bright. We can also take these bright and dim cells and look for differentially expressed genes at the transcript level. And pleasingly, we saw that out of 11 different genes that are known to be differentially expressed in these cell types, based on the literature, we see 10 that move in the direction we would expect them to move. A couple of technical points about CITE-seq. So we wanted to show how it compares to flow cytometry and CyTOF in terms of protein detection. And so if we took the same cells and performed CITE-seq and flow, just in terms of does it detect the same cells in the same proportions, the answer is yes, it does. They're basically indistinguishable in terms of B cells, T cells, non-T, non-B here, just very simple CD19 versus CD3 representation. And if we do the same analysis with CITE-seq, we again see essentially indistinguishable detection of cell numbers. And I'm just going to show for this example, this is the CyTOF work here especially, and a lot of this has been done with Adi Rahman, who's doing a lot of really cool work on CITE-seq and CyTOF and flow cytometry, independent of us. The other question is, how does the dynamic range of CITE-seq compare to that of flow cytometry or CyTOF? And so to address this, we did an experiment where we took cord blood cells again, stained with a 50-50 mix of an oligo conjugated CD8 and a fluorophore conjugated CD8 antibody. We then did FACS and sorted into 4 different bins based on CD8 expression, shown here in these 4 different colors, and reanalyzed these fractions either by flow cytometry or by CITE-seq. And this was, in the early days of CITE-seq, this was done with DropSeq with a relatively small number of cells. So the plots aren't as close as they are for flow, aren't as tight as they are for flow cytometry, but you can clearly see the relationship that we gated for is maintained by CITE-seq detection as it is by flow cytometry. So we're really confident that the detection levels, that the dynamic range that we get from CITE-seq is really comparable to whatever else is out there. For those who are used to looking at flow cytometry data as well, this is a typical representation where you can do these biaxial plots to do pairwise comparisons of 2 different markers to each other. And here's an example from BioLegend, just showing CD4 versus CD8, and you can see cells that are CD4 positive and CD8 positive, and very few that are dual positive. We can see the same thing from our CITE-seq data here. But of course, as you recall, we also have the transcriptomes of all these cells, so we have a bit more information than just the shape of black dots on a 2D plot, and we can actually now integrate this the RNA-seq data to reveal a few more features of the data. For instance, that it's really the CD8 positive T cells that are highest expressors of CD8 protein, and it's the NK cells that have the intermediate levels, as an example. Because we have multimodal data as well, we can do some interesting things with controls. So in this experiment, we spiked in a small proportion of mouse cells into the cord blood mononuclear cells, and we did this because all the antibodies that we used on our panel are meant to be human specific. And so we reasoned that any protein that we see on mouse cells, we can call that background levels of staining. And indeed, doing this enables us to set thresholds and actually cleans up the data quite substantially. We can also, of course, do isotype control type stains, and we do them a lot as well. And of course, these pairwise comparisons, you can very easily generate them for all combinations of markers within your experiment without having to do any pre-gating at all. So, once you do your experiment, in this case, this is, I think, showing 10 different markers, pairwise comparison between all of them. But you can imagine once you start getting up to much larger protein panels, you can get really quite interesting looking plots that you can spend all day looking at. So I'm going to move now into multimodal analysis of single cell data. So this is work led by Rahul Satija and his group, in particular Yuhan Hao, who's a new, relatively new postdoc in Rahul's group, who's really taken this work and ran with it. I should say at this point that our group and Rahul's group have collaborated over a significant number of projects, and we really have this ongoing productive collaboration it's really fun. It's really fun to work with him and his group. So on the top here is the same data set that I've been showing in the previous slides, clustered by RNA. In this case, it's just shown in a UMAP representation as opposed to the t-SNEs on the previous plot, so that's why it looks a little bit different. And on the bottom is the same cells clustered by protein. And you can see you get the same divisions. You probably can't read the labels here, and that's fine. But there's just a couple of points that I want you to take out of this slide. The first one, circled here in 1, is that by clustering by RNA, while you do see the CD4 and the CD8 clusters of CD4 and CD8 T cells, they're really not that separated. There's some intermingling between the two clusters, and it's not clear where the boundary is, as opposed to when you look at clustering by protein, when obviously the CD4 and CD8 protein expression drives a lot of the separation, and you have very distinct, discrete clusters. The second thing is that you can see red blood cells here when you cluster by RNA. You don't see them at all clustering by protein, and that's partly because our panel was small enough that we didn't have any red blood cell specific markers here, but I think it serves the purpose nicely for what I'm about to show. So Rahul and Yuhan were trying to figure out a way to methodically look at joint information of both RNA and protein, realizing that the work we've done previously has just been essentially clustering by RNA and then just seeing where the proteins are expressed on top of that, that we might be able to get a bit more information if we could inform the clustering by both the RNA and the protein. So they took an approach where, just sort of explained here in the next few animations, where if you have a target cell shown here in blue and you look for the nearest neighbors of that particular cell, and I'll blow this up a little bit here so you can see a bit more clearly, and you can see what happens when you consider RNA neighbors versus protein neighbors. And in the case of this CD8 positive T cell here that's highlighted in blue, by looking at RNA neighbors, you'll get CD8 positive T cells that you'll call its nearest neighbors, but you'll also get some CD4 positive T cells, just because the clustering hasn't really separated them out very well. In contrast, if you look at protein, you're pretty much just going to get CD8 positive T cells. So the protein is probably going to be a more accurate way of determining nearest neighbors for the case of CD8. And this is indeed the case. If you plot the predicted CD8 protein values from protein neighbors against the actual measured CD8 protein levels, you get a really strong correlation for CD8. However, if you look at RNA neighbors, the correlation's still there. It's reasonably good, but there's clearly some points where we get it wrong, where cells are called as being nearest neighbors and they're really not nearest neighbors, and the protein counts are not what they should actually be, are predicted incorrectly. The flip side of this is also true if you look at hemoglobin A, going back to the red blood cell example from a couple of slides ago, where if you look at RNA neighbors, you see a great correlation. But if you look at protein neighbors, again, there's a lot of error in this, which again is not unexpected. And so really the question we need to ask is, how important is each modality for each cell? And the way that Johan and Rahul are doing this is that for each individual cell, they're determining cell-specific weights. So basically, for each cell, how important is the RNA and how important is the protein, and placing a number on that and then applying those weights to derive a joint representation of the data. So this is the same plots from the previous slide, again showing the CD8 protein predicted from protein for CD8, protein predicted from RNA, and the inverse for HPA. And again, the ones highlighted in green here show a nice prediction, a nice correlation between predicted and actual. The ones highlighted in red show where this falls down. But the joint reconstruction, pleasingly, can really reconstruct both modalities. And so this is hopefully giving you a flavor of how Rahul and Johan are doing this to sort of do a more formalized joint clustering. And then going back to this same data set from our paper a couple of years ago, the joint clustering, we hope we can show improves classification of cell states. And so again, going back to the previous example, you can see here, this is the NK cells, this is CD8 protein in both the T cells and the NK cells. If we now go to the joint clustered data, exactly the same data, you can see the CD4 and CD8 cells separate out really nicely. So that's really clear that they're clearly distinct. But importantly, the NK cells separate by CD8 high and low. So if you look here, you can see a subcluster forming within the NK cells that is marked by high CD8 expression, and it's distinct from the CD8 low expressors. In the case of our original analysis, these were all completely intermingled with each other. So this is a nice demonstration at the level of 13 protein markers. Where we're moving now is to perform CITE-seq with really large protein panels, and I think this is a good time to really point out. I'm going to show a little bit of data from this. There's actually a huge amount of work that's gone on behind the scenes here that the data is never likely to see light of day, but the optimization and the just figuring out how to get all this to work. And this has been led by Marlon and Stephanie from my group and Bill on the wet lab side, Bill Malk from Rahul Sathija's group. Johan and Shuei have been doing a lot of the analysis work in Rahul's group. Again, this is a close collaboration between our groups, but of course I also want to really point out that BioLegend have been instrumental in making this work. I think I looked a couple of days ago and there's 748 different total-seq products on their website. So they've gone from nothing to having that many catalog products available in a very short period of time. And Bertrand and Kit and their teams have really done excellent work in optimizing individual reagents, but also figuring out how to put all these reagents together in a panel that makes sense and is able to give even representation over a large number of measurements. So I wanted to get that out of the way first, but thank you to all these people. So if we now look at what the data looks like when we do these large protein panel experiments, shown here is a plot of 87,000 human PBMCs from 4 different donors. This is 220 antibodies, all stained simultaneously from BioLegend. These are total-seq A antibodies run on 10x Genomics v3. So total-seq A antibodies work very well on 10x Genomics v3. So I'd like to point out that if you look at clustering by RNA or by protein or by joint, you get a good number of clusters in each representation. I guess just from a sort of 1,000-foot view, you can start to see that the clusters sort of merge together in the RNA and in the protein sets, whereas you can see they really are, in the representations. Whereas when you do the joint representation, you can start to see really quite discrete clusters forming. And this is that same representation from the previous slide, just expanded out and each cluster colored differently. And we're really confident in these clusters because each one of them has a clear expression differences between marker proteins and RNAs. And so we certainly don't think this is a result of over-clustering. We think this is actually demonstrating the true diversity in the system. And the diversity is really quite stunning. So if I reveal what each cell type, what each cluster is, you can see that now we get very clear separation between CD4 and CD8 cells, but within each cell type, there's clearly a lot more complexity than just, is it a CD4 or a CD8 positive T cell? And we're seeing huge numbers of subclusters within these larger categories. And the same thing goes for NK cells, B cells, dendritic cells, monocytes, etc. I should say our panel is more focused towards T cell markers, so we do see more separation in those cell types. So I'd just like to go over a couple of examples now of how this joint representation really aids in getting more information than we can get from RNA or protein alone. And the first example here is looking at Tregs, where this nice cluster of Tregs that we can highlight here, we only see if we do joint clustering. Really, we can't separate these out by RNA or by protein alone. And you can see probably why this is the case is that although CD25 nicely highlights these cells, both FOXP3 and Icaros, two very canonical markers of these cells, their expression is clearly enriched at the cluster level, but at the single cell level, it's not strong enough. So the RNA signal is present, but it's not strong enough on its own. Another example I want to show is this expression of integrin CD49 alpha, which is expressed on a subset of T cells, and it's usually associated with tissue residence, especially in the skin. And if we look at RNA representation or protein representation and look at CD49 protein expression, it's all over the place. It's scattered out throughout a bunch of different cells. However, when we do joint representation, we really clearly see two distinct clusters here, one in the CD4 group of cells and one in the CD8 group of cells, which we don't know what these are, but they're potentially interesting, and it's nice to see how strikingly they pop out with the joint representation. So to summarize this part of the talk, really the concept behind CITE-seq technology is that these oligo-conjugated antibodies allow you to integrate protein detection into single-cell RNA sequencing. And I hope I've shown that if you integrate protein and RNA measurements, you can greatly enhance cellular phenotyping. And this is particularly the case once you start getting into really large protein panels. So for the rest of the talk, I'm going to talk about other applications, how else we can apply CITE-seq technology. So firstly, I'm going to speak about hashing. So this is a method that this is work that was led by Marlon from my group and Shuei from Rahul's group. And it's a way that we can do antibody-based sample multiplexing. So this work was really inspired by a nice paper from Jimmy Yee's group a couple of years ago, where they used genetically distinct donors and multiplexed them together in the same 10x experiment, and then used expressed SNPs, so variation that existed between the different samples, to demultiplex the cells and figure out which cell came from which patient. The advantage that they showed here is that you can from a per experiment cost, this can really reduce that down, because if you only need a couple of thousand cells, then you can put a whole bunch of different patients together and get that many cells while only spending the money on the reagents once. What it also enables you to do is to really confidently identify doublets. So if you see the signature of having two different types of cells in one with sharing one same cell barcode, then you can be confident it's a doublet and you can discard that from your analysis. So partly inspired by Jimmy's work, but also realizing that this type of approach using an exogenous tag has been done especially in CyTOF with CD45 barcoding. We decided to do the same thing for using our oligo-labeled antibodies. And this is just a rundown of how hashing actually works. If you imagine taking three different samples here, you label with hashtag A, B, and C, pool the cells together, and then when you encapsulate them into different droplets, and if they look something like this, you library prep, sequence, you analyze and demultiplex, and then you get something like this, where you look and see what the dominant hashtag that you see in each cell is, and then you can basically demultiplex the cells based on that. So then you can also, of course, robustly identify a proportion of doublets that's related to the number of samples that you've multiplexed together and remove them from the analysis. And so what this enables you to do in our hands is get up to 20,000 single cells from a single lane of 10x genomics sequencing, which is a significant improvement on what you can get otherwise. So this is what the data looks like if you take this experiment. So this is 8 different genetically different donors, each hash with a different hashtag. And then if you cluster by the hashtag expression, you see 8 major clusters as you'd expect, but you also see 28 distinct doublet clusters as well, which is exactly what you'd expect from collision of 8 different samples. The samples all consist of the expected populations by RNA, which is good. It's not affecting the RNA expression, and we've done some pretty extensive tests to make sure that we're not affecting the sensitivity of the systems, and we don't see any drop in UMIs or gene counts per cell with hashed versus non-hashed samples. And the hashtag signal is really highly specific. So if you look and see here, you can see that cells either have the signal or they don't, and you can very clearly see when a cell is a doublet as well, and what doublet it was. We're really pleased to see that we had a very strong agreement with genotyping based multiplexing. So with Demuxlet from Jimmy's group, we designed our experiment so that we could test this out, and we're pleased to see it really did work nicely. And finally, cell hashing is also really useful in that it enables the identification of low quality cells. So we use the hashtag counts as an orthogonal measure of what actually constitutes a cell. And so you can imagine if you have a cell that's really poorly expressing RNA, it's still got a membrane that's still full of surface proteins, and we use antibodies directed against a cocktail of highly expressed cell surface markers, and currently we use beta-2 microglobulin and CD298 mixed together and labeled for hashtags. And so if you take cells that have, I think in this case it was less than 200 UMIs, and separate them out by those that have a strong hashtag signal and those that do not have a strong hashtag signal, and then force them back onto the cluster from the higher count UMI cells, we see that those that actually have a strong hashtag signal look like cells, whereas those that don't really look like the soup or the ambient RNA that's in the solution. The final application I'm going to talk to you about today is a relatively new method that we've worked on called ECCITE-seq. And this is work that's been led by Eleni Mimitu, a senior research scientist in my group. So let me move to the next slide. So to give a general background here, most methods that people are familiar with single-cell RNA sequencing generate 3' tags. So this is where the reverse transcription primer has some sort of a barcode. Upon RT, you make a copy of make a first strand copy of the mRNA and amplify the cDNA by one or by a couple of different methods, and then at some point you fragment this down and make a library that you sequence on an Illumina sequencer that retains the barcode that came in on the RT primer, and then it has enough sequence information from the transcript to tell you which gene was expressed. And so this is how it works for 10x 3', DropSeq, InDrop, DD-seq, and other plate-based methods as well. With 10x's 5' kit, they essentially switch this around. So instead of on the 3' kit the RT primer coming from the bead and the template switch oligo being free floating in solution, for the 5' kit it's the other way around, where the template switch oligo is associated with the bead and the RT primer is free floating in solution. So, and then once you amplify the cDNA, you break it down, you make a library, you now have a 5' tag gene expression library. But you can still use that to quantify transcript expression. But the other thing it enables you to do is for 5' variable genes to specifically amplify and make libraries from. And this is especially useful when trying to decode the paired expression of T cell receptor and B cell receptor transcripts in immune cells. And so how this works is that these transcripts have highly variable 5' regions that start with a large number of V genes, and then there's all sorts of hypermutation and rearrangements that form the D and the J segments, so the choosing of the D and the J and the non-homologous end joining and sort of the messiness that results in this structure here. That's all part of generating the huge diversity that's in the immune system. So once you have this cDNA library, you can then amplify with with constant region specific primers heading back towards the 5' end of the transcript. And now that you've placed this common handle on the 5' end of every transcript that also retains your cell barcode and UMI information, by amplifying this up and then fragmenting down and sequencing it, you can reconstruct the paired expression of these transcripts in cells. So this is what our reagents look like for 3' CITE-seq and cell hashing. In a nutshell, the poly-A sequence here for capture, a barcode, and then a handle. We typically use a different handle for CITE-seq and cell hashing so that we can separate the libraries. And for 5' CITE-seq and 5' cell hashing, they're functionally similar. They have the same handle sequences. Barcodes can be whatever you like them to be. But instead of the poly-A sequence, we have now a region that's complementary. It's a 13 nucleotide region that's complementary to the switch region of 10x's template switch oligo. And so upon RT, this oligo here gets extended, and you now have the joining of the cell barcode and UMI from the bead to the barcode that comes from derived from the antibody. And so Eleni made a panel of 49 different antibodies with all differently barcoded, as well as a set of hashing reagents. And we went ahead and looked at PBMCs from a healthy donor and from a cutaneous T-cell lymphoma patient. And long story short, the CITE-seq component worked nicely. You can see differential expression of, so here CD8 and CD4 in different populations of T cells, for instance, just stands out. But we also have, we can also attain clonotype information, such as TCR alpha beta over here and TCR gamma delta over here. And this is useful when looking at this type of cancer, because you can do things like this, where we take the two different samples and align them together, so we can see the control and the CTCL patient here. We can see which cells have a productive T cell receptor, and fortunately and pleasingly, these overlap with cells that are expressing the CD3 protein, as you'd expect. But then we can separate these further into clonotypes. So shown here is the top clonotype in the CTCL patient, which is obviously greatly expanded compared to the top CD4 positive clonotype in the control, where there's just a smattering of these cells around. And so we can do something like in silico gating here, right, where we take cells that are both CD3 and CD4 positive, so CD4 positive T cells, and just analyze these cells only in the control and the CTCL. But then we can further separate them out by which receptor they express. And so if we do this, we can compare at a pseudo bulk transcriptome level the control CD4 cells to the polyclonal CD4 cells from the CTCL patient to the monoclonal CTCL malignant cells. And you can quite clearly see there's a very strong gene expression phenotype associated with transformed cells, as you'd expect. There's another application of this CITE-seq, ECCITE-seq technology I'd like to briefly talk about today. This wasn't in our original paper, but this is something that we've been working on with Nick Skourakis' group at UCSD. So Nick's come up with a really cool, or Nick's group has come up with a really cool way of generating peptide-loaded MHC tetramers using a chaperone-mediated exchange, which actually makes this whole process really quite cheap and relatively easy, in their hands at least. So we've worked with him on making this such that it can be read out by a sequencing-based assay. So just to remind, this is what protein tags look like from ECCITE-seq, in this case conjugated to an antibody. For a tetramer tag, we put a different handle on so that we can recover the library separately, but you can imagine now this tetramer is binding to the specific T cell of interest, and you can pull that down and sequence. And of course we've worked with tetramers so far, but there's no reason this method can't be installed onto a different backbone, like a dextran backbone, to create dextramers. And so in terms of just proof of principle, this is showing the very sort of well characterized MART1 DMF5 pair, where the MART1 peptide is very strongly and specifically recognized by the DMF5 T cell receptor. So cells that are expressing the cells here are ordered based on their levels of expression of DMF5 receptor, and I think it's hopefully you can see that cells where we detect DMF5 receptor are also the same cells where we detect MART1 tetramer. And this can obviously then be done in combination with hashing and CITE-seq and transcriptome, and so you can get all this different information. You can recover the TCR and also the epitope that it's binding to. And yeah, this is work led by Nick Skrakis' group, with particularly Sarah Overall and Joey Tor. So the final application that I want to talk about of ECCITE-seq is in direct guide RNA capture. And so, as I mentioned previously, it's now with the 10x5' kit, it's the template switch oligo that is appended to the gel beads, whereas it's the RT primer that is free floating in solution. You just add that from a tube when you set up your reaction, and there's no reason you can't add some extra primers as well. And that's exactly what we did. So, different primers here that will bind different things with a different handle if you want. And we reasoned that a really good candidate to start with here was to use this method to directly detect the Pol3 generated guide RNAs for Cas9. They have the structure that lends themselves to this. It's variable at the 5' end, where the region that actually targets Cas9 to particular genomic locations, but the 3' end is invariant. And so our goal was to make a method that would enable us to do CRISPR screens with a single cell multimodal readout. And so methods have been published to do CRISPR screens with RNA-seq as a readout. The CRISPR-seq and Perturb-seq sort of illustrated here on the left-hand side, and crop-seq on the right. Both of these methods rely on a proxy, so a Pol2 generated mRNA that gets captured along with the cell's mRNA and reads out the identity of the guide that was present in that particular cell. In the case of CRISPR-seq and Perturb-seq, this takes the form of a guide-associated barcode sequence that exists in the vector at some distance away from the guide itself and is expressed on a separate transcript. In crop-seq, using sort of a trick of lentiviral biology, the guide RNA expression cassette is cloned into one of the LTRs, and during integration is duplicated and then becomes part of a read-through transcript from the antibiotic resistance gene. In particular, on the left-hand side here, the CRISPR and Perturb-seq methods do have had some documented issues with switching between barcodes and guides, where the barcode that you think is associated with guide ends up not becoming associated with that guide because of some template switching going on. And so we're sort of motivated by, I mean, these are both great methods, but we were motivated to maybe try and take advantage of the fact that we have this, the molecule that's actually doing the perturbation is present at very high copy number, and we reasoned if we could capture that directly, then we could use this method to use existing existing libraries, and probably have a very high capture rate of these transcripts. So this is what the data looks like. If we take, this was just a very small scale proof of principle screen where we took 20 different guides against targets that we were interested in. We were really pleased to see that we could detect the guide in greater than 98% of cells. And also by doing this together with cell hashing, we could discriminate cells that were, we could discriminate doublets that were a result of two cells being partitioned into one droplet from doublets, well, apparent doublets where we had two guides per cell as a result of two different viral particles getting into that one cell. So now if we cells clustered by guides here on top, you can see very clear clustering, separation of different populations. And looking at the mRNA level, there's, in most cases, you can see an apparent downregulation at the transcript level specific to the clusters expressing the guides targeting that particular transcript. And where we have protein reagents available, in the case of CD29 here, ITGB1, and CD46 here, you can see downregulation at both the protein and mRNA level for these particular in these particular clusters. So there's another way of looking at this data represented on the next slide here, where we order cells based on their levels of guide expression. So this is the number 2 guide for CD46, and you can see from left to right here is the expression of the guide. And I hope you can see that I think this actually is a nice illustration of just, it illustrates a few nice features of protein detection as illustrates the strengths of protein detection, where if we look here, you can see this cloud of dots. So each individual dot here is the protein level in a particular cell. And you can see it's very robustly detected at the protein level. There's very few cells that have 0 in the absence of the guide. However, once the guide is present, all these dots collapse down to the very bottom here, indicating we've got very strong knockdown, but also just nicely illustrates how robust the detection of protein is. When you contrast that to mRNA, where we know that this gene is expressed in all of these cells, but there's a whole bunch of cells here where we never detect this mRNA in the first place. And that can also be reflected in the output of the screen, where even essentially you see the cloud of dots here just getting a little bit darker here and maybe a little bit more sparse up here, but it's not the same reduction from all to nothing that we see at the protein level. Another way of looking at this is seeing how many cells we need to look at randomly before we see a differential gene expression for this particular target. And so if we look at the 3 different guides shown here, 1, 2, and 3, at the protein level, you can see that, and this is the p-value here of differential gene expression, you can see it, even with a very small number of cells, it's really apparent very early that we detect differential expression as a result of the guide being present. Whereas at the mRNA level, it's really only with one guide, the best performing guide, that we see significant differences, and even then it takes quite a number more cells in order to see that. This is clearly just a proof of principle example here. This is not the sort of screen you would typically do, but what we think is that coupling this type of, coupling this fact, the fact that you can detect protein more robustly and you can detect changes more robustly, with the ability to really ramp up the number of proteins you can interrogate in a single assay, as shown by our 220 panel experiment a few slides ago, earlier in the talk, we think that the gene expression signatures that people use as readouts for these CRISPR screens will just become so much more robust with significant amounts of protein markers in there as well, which will hopefully give stronger data, but also just enable you won't need to interrogate as many cells for a particular perturbation, which will then hopefully have the flow-on effect of being able to screen more perturbations in a single assay. So just to summarize ECCITE-seq, so this is expanded CRISPR-compatible CITE-seq. We're able to capture a bunch of different modalities using just the standard 10x kit. From the cDNA, we can get gene expression, TCR, BCR. From oligo-labeled proteins, mostly antibodies, but also MHC tetramers, we can detect differentially expressed proteins. We can do sample multiplexing through hashing. We can also detect MHC tetramers, and really we think any binding event, any binding event that you can stick an oligo on and still have that protein or whatever the molecule is binding to its target, we think is probably detectable by this type of method. And with respect to non-poly A RNAs, we've shown we can really robustly detect guide RNAs, but we think this approach can be extended to other non-poly A RNAs, and we can imagine putting in 100 different specific primers to genes of interest. So to summarize the whole talk, I hope I've shown you that by integrating protein and RNA measurements, we can really strongly enhance cellular phenotyping. I think hashing is a really useful tool for multiplexing and cell overloading. I didn't mention it earlier, but there's a bunch of other methods out there around hashing, not necessarily using antibodies. There's nuclear hashing that is using antibodies from Jellick Goldblum and Aviv Regev's group. There's some really nice work using lipid-tagged lipid-tagged oligos from Zev Gartner's group called Multi-seq. There's some direct conjugation of oligos, transfection of oligos. There's a whole bunch of methods out there around the multiplexing of single cells. And you should check out all these papers, they're all really good. So CITE-seq and hashing are both now compatible with 3' and 5' 10x technologies. we think we've shown a nice example where if you use TCR reconstruction together with CITE-seq, you can really interrogate a complex cell population in a really interesting way. And finally, CITE-seq shows that you can capture at least 5 different modalities and likely more in a single assay, including direct guide RNA capture for multimodal CRISPR screens. I just want to leave with a quick plug. We have a website that we keep updating. This is CITE-seq.com, where we post protocols, tips, tricks, FAQs. We have a contact form on there. So if you have any questions, if you want to run CITE-seq, please feel free to contact us. We really do try and answer most emails in a timely manner. Finally, I'd just like to thank my group, the wonderful group of people to work with. I've mentioned several of these people already. Marlon, who's now at 10x Genomics, really started the CITE-seq work. Eleni's continued it with ECCITE-seq. Stephanie has been really instrumental in the hashing work, in the MHC Tetram work, and in the very large panel work. I haven't had a chance at all to talk about the work of Kunal, Will, and Chow, but they're also doing interesting work some in single cell space, some in other genomic technology development. We're partly funded by Chan Zuckerberg Initiative, by the NHGRI. I want to thank BioLegend, really especially BioLegend. The work that they've done in making these panels has really been instrumental, and I think I'm really excited to see where this goes in the future. Brian Fritz from 10x Genomics helped us out with some free kits to get ECCITE-seq working initially. Want to thank my collaborators. I've mentioned Rahul extensively already, but we really do have a great relationship with his group and continue to. The ECCITE-seq work was together with Sergey's group, Neville's group, and Jen Qing's group. And of course the work that I mentioned from Nick Skarakis' group on the tetramer work. And finally, we're hiring. We're looking for new research scientists, so if you're at all interested, please reach out to me soon. And with that, thank you very much, and I look forward to taking your questions.
Let me ask you that first question. 10x says they don't support TotalSeq-A for their version 3 kits. Does it work?
It does. It's so TotalSeq-A has a poly A tail, which is designed to interact with poly T primers. 10x's V3 kits have a separate technology that they've invented called their feature barcoding, which has a different handle, and so they have a separate set of CITE-seq-like reagents, TotalSeq-B reagents that are complementary to that. So I think that they're sort of trying to push people towards using those reagents. But we use TotalSeq-A, and it works fantastically well in our hands. There's no detrimental effect on the mRNA quality, and yeah, so we think either can work just fine.
Okay, the next question is the audience member starts out by, please ignore my ignorance, but I'm curious, which software do you use to analyze your data? Do you have experience with FlowJo's GenSeq software? And which software is the most user-friendly for a flow expert but known to sequencing? Maybe new to sequencing?
Good question. So we most of what we do is in Seurat. So this is very commonly used software in single cell sequencing space. This is developed by Rahul Satija, who I've mentioned several times during the presentation. And that would be probably, familiarizing yourself with that is probably the best thing that you can do, especially given the work that he's been doing with integrating RNA and protein together. There are, however, other options. 10x Genomics has a very user-friendly software called Cell Ranger, which is completely free. That can use both protein and RNA data, and you can do all sorts of nice, cool manipulations. For people who are used to using flow software, SeqGeek is something that's been put out by FlowJo recently, which will integrate this sort of work. I'm not entirely sure that it yet supports protein tags, but it may. And we've also seen work by Partec, who are also putting out software that is useful for detecting, for looking at this type of data, but single-cell transcriptomes and proteins.
All right, Peter, another question. Is CITE-seq compatible with systems other than 10x Genomics?
Yeah, so we've run it. We initially developed CITE-seq on DropSeq. This was, we started working on this before 10x had a single-cell offering. And then we quickly, obviously, adapted it to 10x. The poly-A, the TotalSeq-A type reagents will be compatible with any method that uses poly-T to capture cells' mRNAs. So we've personally used them on Bio-Rad and Illumina's DD-seq. I'm not sure how much that's being used these days. And we know people who are using it in InDrop as well, and we've done plate-based type methods as well. So yeah, I think long story short, any method that's going to that's going to use poly-T capture beads or any other type of poly-T capture, CITE-seq can easily be made compatible with.
Okay, thanks for that. The next question is, how do you perform titrations for CITE-seq?
Good question. So this is something that we spent a bit of time carefully, I guess is the answer. This is something we spent a fair bit of time on earlier. The problem is it's quite an expensive thing to do. So actually, this was part of the motivation for developing hashing in the first place, was that we could do a bunch of titrations and put them all together in the same experiment and the same sequencing run. So yeah, we would do titrations in the same way that you would normally do titrations. You'd start with a range of concentrations and see at what point your signal to noise gets to the point you want it to be at. There's another thing that you need to bear in mind with this though, which is, I mentioned that flow cytometry, for instance, you're measuring different fluorophores in different channels, so there's different excitation and emission spectra. So if one fluorophore is 100 times brighter than another, it doesn't matter because you're measuring them independently. With respect to sequencing, though, if one signal is 100 times more prevalent than another, you're sequencing everything in the same channel effectively. So if you do have that sort of situation, then you need to sequence 100 molecules of molecule A before you get to one molecule of molecule B. So it can be a bit harder to get the sort of dynamic range that you want. So beyond performing titrations to achieve optimal signal to noise, we found that when constructing large panels, it's really important to get balanced panels as well, which can sometimes mean making sure that we don't have some very dominant marker completely taking up all the sequencing space. But I guess in terms of actually doing titrations, we do them as much as we can. These days, we seem to be working a lot more with BioLegend, who are doing a huge amount of work behind the scenes in actually getting the concentrations right and working on developing panels that are pre-titrated. So hopefully a lot of that will be taken out of the hands of the actual experimenters at some point in the near future.
All right, Peter, I have, I think, 4 or 5 more questions for you. So
Sure.
How are CITE-seq and hashing antibody-oligo conjugations performed?
So in our, when we did it ourselves, and we are by no means protein biochemists, we initially did some streptavidin and biotin conjugations to attach the oligos to each other. And then we moved to a direct conjugation chemistry with using click chemistry. And this protocol is on our website. It's also present in our cell hashing paper. But as I mentioned, and then we did some sort of crude purifications to remove unbound oligos. So I'd refer you to those papers if you wanted to do it yourself. We have certainly found that the reagents from BioLegend seem to work better than our own reagents. They have much more control over the site of conjugation on the antibodies, whereas we don't really have control. When we conjugate, at least the method by which we do it, it can conjugate at any point on the antibody, which can sometimes lead to an antibody failing. Sometimes, yeah. But we've certainly had better success with the antibodies from BioLegend. This is kind of what these guys do.
So, Peter, we're running out of time. I think I'll ask you maybe 1 or 2 of those questions, and the other ones we didn't get to, we will answer via email later. Which antibodies do you use for hashing?
So we worked together with BioLegend on this. So I'm not sure if I covered this very well in the actual talk and listening back, but it's important that you try and get antibodies that will decorate every cell. For hashing, you really don't want to discriminate one cell from another. You want every cell to have an even staining. And so in that case, for human cells, we use a mixture of CD298 which is the sodium-potassium ion channel, and beta-2 microglobulin, which is part of the MHC complex that is, again, present on every cell. Now, we do see that, in certain, it's a known fact that cancer cells will often downregulate MHC so that they're invisible to the immune system, and so this does lead to issues where you can see less cells. Cancer cells may not express the at least B2M very highly, and so that's where having a mix of different antibodies in there can sort of ensure that you get staining of your whole population.
Okay, Peter, I am told that we have more time than I thought, and I'm also told that we just got a bunch more questions in the inbox. So I don't know how game you are to answer many more questions, but if you want to take a look at what we just got and see if any of them are priorities that you think we should address, or I should just keep going through.
Just ask. It's totally fine. Yep. Please ask whatever you've got.
Okay, so let me just start with this one because I have it up now. How many cells can you load with cell hashing?
Well, you can probably load a very large number. We've found that the sweet spot is to recover about 25,000 cells, of 25,000 productive cell barcodes from a lane of 10x. Now, what this means is that about 5,000 of them will be doublets, and you'll be left with about 20,000 single cells that you can confidently say are single cells. And so this is using 10 different hashtags. You could use more hashtags if you want to, but the number of doublets that you can identify is directly related to the number of hashtags that you use. And so in our case, when we're trying to get as many cells as possible for an experiment, we typically use 10 hashtags to stain our sample or samples, and we recover 25,000 cell barcodes, which ends up giving us about 20,000 single cells.
Okay, here's one of the new questions that came in. What will the standard procedure to test the best condition of CITE-seq for new antibodies?
Standard procedure to test the best condition of CITE-seq. For a new antibody, that's a good question. So we haven't done a huge amount of work with new antibodies. Most of the time, we're working with antibodies that have been very well characterized by flow cytometry or by CyTOF for a number of for many years and pretty much well accepted. What the advantage that CITE-seq does give you is, you probably have a pretty good idea which cell type you're expecting your antibody to stain, and you also the protein target the antibody is going for, but you also know the transcript of the gene that is producing that protein. So to be confident that it is staining what you expect it to stain, I would say you would hope to see it enriched in a cluster that is also enriched for the transcript that you're interested in. Best case scenario. I think that's probably the best advice that I would have. But assuming that you've already tested your new antibody to show that it's specific. You've done appropriate Western blots and controls, or maybe not Western blot, flow cytometry controls. If you can get a cell that's knockout for that protein and show that the staining disappears, I think that's probably the best advice I can give.
All right, so Peter, it turns out the next question wasn't a question. It's a statement from Patrick at BioLegend. He wants people to know that the total seq-A antibodies are definitely working on version 3 of 10x.
Thank you, Patrick. We agree. I hope that was clear in the presentation today.
All right, so now I have another multi-part question, so get ready.
Sure.
For CITE-seq, to subtract background, is it better to use mouse cells compared to isotype control? Have you tried clustering with protein and RNA in cells that are undergoing RNA decay or some sort of RNA regulation? And could you be missing information here? How about cells that are undergoing splicing? Did you look into which isoforms your antibodies recognize? Thank you.
Okay, so fortunately I have this one in front of me, so I'll address each part at a time. Mouses versus isotype controls. Early on, when we were doing the work ourselves and conjugating ourselves, we found the mouse cells to be really handy. More recently, the isotype controls seem to do a really good job. I mean, I think this is just a fact that the BioLegend antibodies are cleaner than the ones that we were making ourselves. So, I mean, I think practically speaking, it's a lot easier to just include a few isotype controls in your experiment than it is to maintain mouse cells that you spike in at the right concentration. So probably isotype controls are an easier way going forward, especially if you're using antibodies that you know that have been very well characterized, that are human specific, etc. Then you may not necessarily need the mouse control. The next part, clustering with RNA undergoing RNA decay or some sort of RNA regulation. Not specifically. So sorry, I should repeat the question. Have you tried clustering with protein and RNA in cells that you know are undergoing RNA decay or some sort of RNA regulation? We haven't specifically targeted cells doing that. That this is very clearly we could be missing some sort of information here. There's some very interesting work going on in looking at RNA velocity and things like that, and I think actually Leo Pachter's group talked about put something out about RNA acceleration recently, which takes velocity and then includes protein data as well. So yeah, I think there's probably some very interesting information there to be gained. It's not something we've specifically looked at. With respect to cells that are undergoing splicing, I guess we mean alternate splicing. I think splicing is pretty universal. Did you look into which isoforms your antibodies recognize? In most cases, these are very much mapped epitopes, so it is very well known which isoforms these antibodies will recognize. These are all monoclonal antibodies. But you can certainly use an example like CD45 RA and RO. These are two different splice isoforms of the CD45 protein. CITE-seq can be very readily used to discriminate these markers from each other. It works very nicely. Whereas obviously at the transcript level, it's very hard to do that. You may not get enough reads in the exons that discriminate those two isoforms. Okay, is that okay? I think I covered.
You got that covered? Okay,
yeah. Good.
So I'm just going to ask you two more questions, and then we'll wrap this up. Are there any effects of staining with CITE antibodies on mRNA transcripts? This person is asking if engagement of receptors on the cell surface by the antibody will induce downstream mRNA transcription.
Yeah, I think that's a really good point and good question, and there's definitely receptors can very much be activated by binding of antibodies to the extracellular portions of them. We typically do all of our stainings at 4 degrees and try and keep them relatively short, so we're hoping to not capture too much of a transcriptional response mediated by the antibodies binding, but they certainly could do that. It's not something we've really looked too carefully at. But yes, it's definitely something to bear in mind. It's a good reason to keep the stainings happening at a cooler temperature to slow down any signal transduction events that might happen, and also to keep them as short as possible.
Okay, this is our last question. If we didn't get to your question, please know that we will answer it via email after this event. So Peter, here we go. Would you recommend polyclonal antibodies over monoclonal antibodies for conjugating to oligonucleotides?
No, I think the monoclonal antibodies, if all you have is a polyclonal antibody, then you give it a try for sure. I think monoclonals are typically, depends on the source of the antibody, but most antibodies that are produced by Legend or any other company out there are monoclonal. That's very beneficial for a number of reasons. It's a well-characterized binding event. There's no batch-to-batch variation, or there's very little batch-to-batch variation. And the purity of the monoclonal antibodies is typically very high compared to polyclonal. So no, if you have the choice and you have a functional monoclonal antibody that can give you the same answer that the polyclonal you have, then I would go for the monoclonal every time.
Again, I just want to thank Peter for joining us today and sharing his knowledge, and a huge thank you to BioLegend for sponsoring today's event. We're going to send an email in about 24 hours advising you that the webinar is available on demand, and telling you can share it with colleagues, etc. But we really appreciate you joining us today, and we look forward to seeing you at our next event. Thank you.